All right. Well, hi, everyone. Welcome to our Industry Educator Speaker Series done in partnership between Illumina and Biocom California Institute. This is part of the Illumina Genomic Discoveries program, where we work on bringing education and the industry together in the field of genomics to bring resources for DNA sequencing and the field of genomics to your classrooms. So we are so thankful to have you all here today. We're really excited for a fantastic presentation. Before I introduce our speaker, I do just want to mention that please feel free to ask questions in the chat throughout the presentation, and then we'll ask them at the end during a Q&A session. As well, at the very end, please stick with us after our keynote address. We will share with you multiple resources that we have available to connect what we talk about today and what our keynote speaker discusses today, and how to bring those resources to your classroom, and how to bring this learning to your classroom. So with that, I am going to introduce our speaker. This is Obi Onyeacholem. She is a staff engineer with Illumina Inc., and she is going to talk all about today the work that she does with Illumina and the development that has gone into making a lot of the technology that we use, as well as some fantastic resources. She also see her in the virtual field trip, which we'll talk about a little bit at the end. So thank you so much, Obi, for being here with us today. And I'm going to go ahead and pass it off to you.
Awesome. Thank you so much, Michaela, for that introduction. Let me go ahead and share my presentation. Okay, can everyone see the screen, the presentation? Yes, okay.
Yes.
Thank you. Sorry, my chat window is a little minimized here. But thank you, everyone. Good afternoon. My name is Obi Onyeacholem, as Michaela mentioned, and I currently work at Illumina. I'm excited to be here today to talk to you a little bit about my journey at Illumina, what I do, what Illumina does, and all the exciting work that my team here gets to be involved in. So to start off, I just wanted to give everyone a little background of my career at Illumina and how it has progressed over this now has been over a decade, which always astounding to me to think about that it's been that long. But it started when after I graduated from college. I studied biomechanical engineering and entered the research associate track here at Illumina in the chemistry development department. I was really fortunate to start right off the start to get involved with some of the beta array projects. Those were some of the earlier technologies here at Illumina. And then I started transitioning on to some of the flow cell projects and worked on the bonding technology and then other variations of the beta array technology that we've worked on as well. And you can see as the years progressed, I advanced in my career, as well as got more leadership in various projects that I worked on, including the MiniSeq, iSeq, and most recently the NovaSeq X. So that time point was actually the point in which I started working on the project, but the project had several years before that point and if development and work included in it. But that's where I joined in, and I'll talk a little bit about that project and the other work that I do at Illumina as well. So just a little bit more about me and some of my other interests, both within the Illumina community and outside of Illumina. Well, one, I was born in Cleveland, Ohio. My parents had immigrated there from Nigeria back in the 70s, chose Cleveland, which I get a sense of my choice of choosing San Diego over Cleveland. I wanted to get away from the snow for a little bit, but always still has a special place in my heart. If you are familiar with Cleveland, one of the big staples is sports, and so that was something that I've always been interested in as a young girl, and I participated in a lot of various sports in my adolescence, and continue that at Illumina as we offer some intramural recreational leagues at Illumina, and I've one specifically is softball, so I've been really involved in that league as well as helping to organize it over the last few years. In addition to sports, I've also been very passionate about STEM outreach and volunteering, and so I've been finding ways to work in various outreach programs and initiatives, including Future is Bright with Vanessa and Tiana, I believe, who are on the call, as well as looking into other ways that I've been able to combine those STEM outreach initiatives that I've been interested in our employee resource groups, and specifically Shades, which is a group that focuses on underrepresented populations. So I've been leading that team here as well. So that has been something that has been very near to my heart, and I've been working to try to find ways that we can continue to support those efforts as we move throughout the years. And in addition, I also just love to travel, love book clubs. I've been involved with that with my sister and a few friends, and you can see all the various places we've traveled over the years. And I have a nice little picture too of my softball crew, Illumina softball crew, and other activities at various institutes and schools. So just a little snapshot. If you're interested in any of these things, feel free to reach out. So now diving into what I'm planning to cover for this presentation. I wanted to step through a little bit about the various technology at Illumina and how it's progressed over the years. So I wanted to give a little bit of background there, talk about some of the products Illumina currently offers, and then dive into what I do and how I contribute in the various development projects that we bring forth at Illumina, and then end off with just a little bit of a snapshot into the NovaSeq. So to give everyone a little bit of background and a starting point of how much the field of genomics has progressed, we're going to start off in 2006. So in 2006, what a typical genome center looked like, it included several large instruments. You can kind of see through the picture, there's these massive sequencers at the time, and liquid handling robots, PCR machines, and several other people to support these labs. And what it came down to is that in order to have this type of lab back in 2006, there was a lot of effort to go into it. And in terms of what was actually able to be acquired in terms of data, it was only at that point about 3 to 5 gigabases per month. And so if anyone's familiar with what a human genome size is, 3.2 gigabases. So you can see that at that time and point in 2006, we were only getting about a human genome's worth of data in that year. So, and I'm sure, as I'll cover a little bit more about how much data we are now able to get, you can kind of gives you an idea of how far we've come. Now going into Illumina's portfolio and how over this past now over a decade, how Illumina has contributed and driven a lot of the innovation and data output we've been able to gather. So this gives you the almost full suite of the Illumina sequencers that are offered. You can, as most people probably know, there's a couple others here on the later end that I'll touch on as well. But you can see that coming from where we started back in 2006, one of the first major products at the time, the genome analyzer, we've then worked on that and improved it further to continue on to make these various iterations of sequencers that have allowed us to increase the amount of data that we've been able to gain from these various runs. And we're just working to keep making these innovations more advanced, more developed, so that we can hopefully increase the amount of data and increase accessibility. So this just gives, not going to spend too much time on this slide, but this gives you an idea of, if you were to look at the various, how we phrase it as platforms of sequencers, you can see how they all match up with each other. If you were to break up the various components of these sequencers into just like the chemistry, the optics, and then the flow cells that we put into the sequencers. And these are how they all pair up with each other and how we've been able to and continue to work on the technology of these various products. then now to think about how these sequencers fall in terms of data output. Our iSeq is on the lower end. And so the iSeq, a lot of the main applications for the iSeq include the small whole genome sequencing, which can be, you can think of viruses or small microbiomes. There's also the mid to high throughput, which includes our NextSeq, our MiniSeq, MySeq, and those all give us other additional applications such as targeted sequencing. So if you're interested in a specific part of the genome that you have a reference genome to look into, and you just want to understand like the variants of that specific part of the genome, you would probably go for somewhere in here. And then of course we have our highest throughput instrument. So this is the NovaSeq 600, and we'll also now include the NovaSeq X. And that's where you really dive into the applications that are specifically targeted towards the whole genome sequencing. So when you're interested in obviously human, animal, plant, those are the instruments you're looking towards, and that gives you the opportunity to have a wide range of access to that data that you'll gain from the genomes. And if there are any questions, do feel free to put them in the chat, and we'll make sure to try to answer as much as we can at the end. So just to take a step back into what type of technology and what's the general workflow when you think of sequencing at Illumina. And so there's obviously a ton of information that can be shared about SBS and sequencing, but just to give everyone kind of a visual and general steps of what it looks like and what it includes, you have start with is your sample, so that's your DNA strand, and we alter it by adding adapters. That allows us to then graft onto our surface. And so on our surface, we have what we call primers, and those basically allow you to continue to replicate your various strands of your DNA sample. And so you can see that this will continue on until you have what we call a cluster. And so that's like thousands of copies of this sequence. And ultimately, we go through the kind of like the end portion, which is towards the analysis part, or the detection and then analysis. And that involves an imaging component. And so each one of these bases are going to be fluorescently tagged. And that's what we capture with our cameras and our sequencers. And so we get these images, which shows us what sequence is on the surface. And so we're able to do this multiple times through various cycles and ultimately get to the point of where we have the sequence that we're then can compare to our reference genome and understand what the differences are that may be present. So there's so much more to go that we could dive into this, but in just the interest of this presentation, there'll be a few more slides in the appendix, as well as in addition to the DNA decoded packages that will be offered if you ever want to learn a little bit more about sequencing and some of the other additions to it. So just to kind of give a little bit more of a visual too about how we've been able to progress in terms of cost with sequencing. So when we look at this reference to Moore's law and the idea that, you know, as you double the quantity, you should ultimately have the price. But what we found is that with these various technologies that we've been able to introduce, we've more than exceeded that having a price. And so what's interesting to us is that we can see kind of those really strong inflection points of when certain products have been introduced and how that's affected the general price of the genome, which you don't hear on the left. And so kind of those major points in time have been the GA introduction, HiSeq introduction, and the HiSeq X, or the HiSeq X, as well as the NovaSeq. And so we're just hoping to continue to drive that cost down as we introduce more products in our portfolio. So now that you have hopefully a good understanding of Illumina and the technology, I'm going to dive now into what I do and how I kind of fall into this, you could say, workflow of products. So here's in general what that process looks like when we have an idea, a development project. What are the various steps that we reach in order to get to a point where we launch and provide this product to customers? So just to reference, this is specific to Illumina, but is, I would say, pretty general, can be generally applied to a lot of different biotech companies, but this one is part of our development process. And so where we start is concept. This is this novel idea that our scientists, engineers will think of, that we think will be received well, and we start brainstorming together in small groups. It's usually very, very small teams early on in this concept phase, and they just start determining, is it even feasible? Do we think that this idea that we have can work? And so that's the initial concept phase. And so usually between this point and feasibility, I like to say they show it works at least once, and then present it to upper management to decide, should we move forward? And so that the next phase, once it's decided that this is a possible product or project, is that we go into feasibility and planning. And so then this is where we start really diving in a little bit more into the general planning. How long is it going to take? What type of what amount of support is needed, both from the people to the instruments to the various technologies? Those types of conversations are had in the feasibility and planning phase to really determine then, is this something that we can immediately move forward with and understand how long it's going to take? And so just depending on what maybe the market looks like, resources at the company, this may push it on into development, or maybe it may stay a little bit longer in feasibility. But then once it gets the green light to go into development, that's where we really expand the team, because at the point of development is when you have to further define more of the design outputs, as well as verify the design itself. And so that could include tons of documentation, a lot of experimental planning, a lot of just group meetings about how are we going to meet whatever deadline, which could be anywhere from a year to 2 years, and maybe a little bit more. But also, this phase incorporates a lot of the goals and deliverables that we want to put together, which could be, you could think of it as like data metrics that we want to achieve, data output we want to achieve. Is it a new chemistry we want to introduce? And so all of those are very, very clearly defined during this phase, and then continually tested during this phase as well, to the point that how I say is that, concept feasibility shows it works at least once or twice. Development shows it works at least tens to hundreds of times. And so to that point, once you show that you're able to get something stable enough, as well as something that's able to be scaled up enough, that we can then transfer it over to pilot. And the pilot is this time point is where we start introducing that manufacturing process. So this is the time when we want to start producing thousands of products. So whether it be thousands of flow cells, hundreds of thousands of samples and chemistries, reagent tubes, cartridges, all of the things that go into our various products, and get it to a point that can then be transferred into sustaining. And so at this level, you're a very stable product. It's in the customer's hands, and our team now at Illumina is just monitoring to make sure that we don't see any major failures. And if we do, we're able to address them quickly and hopefully take in all of the feedback that we gather from the customers to improve the product. And so this gives you a general overview of what normally happens once we have a product that is brought forth to our teams, and how we progress to the point of launch. And so where I come in is in that development phase. And so when you think of development is kind of a vague term, like what is development? What does it look like? What's the goal? But to give you a little bit more insight into development, it's a vague term because there are several teams that support development. And this gives you just a quick snapshot of the various teams that can go into supporting the development phase of a product. It can be anywhere from software to user experience to assay research. You have bioinformatics, program management, the list kind of goes on and on. And so there is so many buckets within development. And to think of all of these teams or sub-teams within it, and this can often then result in a team that could be hundreds of people large, that can span many regions across the world and various expertise. And all of these people are working towards this goal of producing this product. And so where I come into this development phase is in consumable and integration. And so what does that mean? So examples of some of the items that I could work on include anything within this consumable bucket. I've more recently have been very much focused on flow cells. And for just a quick background of what a flow cell is, a flow cell, well, an example of one is pictured here. So that's two glass pieces with these fluidic channels that are fabricated into that sandwich of the glass pieces. And on that surface is where we do all of our assays. And so I focus on, myself and my team, we focus on that integration of all of these components of the flow cell. So when you think about the flow cell, you have the material, the glass itself. You have the other surface modifications that we may introduce into the flow cell. You have, that's like more of the chemistry, biochemistry. Then you also have the fluidic channels. So there's a ton of modeling that goes into understanding how to design, say, the width of the flow cell channel, the length, these little like angles and corners, how they may affect the fluidic model within it, and how that's going to affect how much reagents you're going to use, how much of your sample you need to deposit on the surface. All of those things have to be taken into account when you think about these various components of putting together these consumables. And so this just lists kind of a little bit more about some of the examples of other things we could look into, including something as simple as a barcode. You don't know how many experiments can go into testing a different barcode type, or the different types of packaging materials you could have for the flow cell or for your reagents. And so all of these points here are just examples. There's honestly pages long documents of various design inputs, deliverables that we need to test, and all of these things we want to make sure that we vigorously test enough so that when they do get introduced into the customer's hands, that we try to eliminate as many of the potential failures as possible. But I'm going to kind of talk a little bit about, or dive a little bit more into one of these aspects, or one of these deliverables, and that's the surface chemistry shelf life. And so when you think about shelf life, you can relate it to a lot of other products that you may be familiar with, like food, or maybe like cosmetics or things. And all of those consumable products have a shelf life. Like, how long is the time frame in which you can expect to still get a high quality product before you would maybe throw it away or want to replace it. And so we do this with our flow cells and every other consumable product that we offer. And so what our goal is when we start thinking about evaluating shelf life is that we want to make sure that with whatever time frame we have, what we're able to test the worst conditions possible, so that we're confident in the general performance in the field. And so we can go through these various buckets of experiments. And so an example, real-time shelf life, that could be putting the flow cell in the expected storage condition, which for most of them is 4C in the various packages that we provide, and putting it, say, in that fridge for a whole year, and then taking the flow cell out after that year and putting it on the sequencer and seeing what type of data quality we're able to get out. So that could be an example of one. Another way that maybe if you have a shortened amount of time, we may look at accelerated shelf life. So that is based on an equation in which we are able to convert by how much we increase that standard storage temperature, and then compute that to what that equates to a specific amount of time. And so that allows us to hopefully get answers in a short amount of time by exposing it to these various high temperatures. And then in addition to that, you have the areas of reagent stress. So I mean, reagents have their own whole bucket of different ways that they may be interested in testing shelf life. But one way that we often combine or integrate the two is we take that reagent in its stress condition, put it on the flow cell, and then test it. And so all of these various components we want to have at their worst case or at that edge of poor performance, test it all together and make sure we're still okay. And then usually we put like the shelf life maybe 3 to 6 months before that time point, so we have enough of a buffer when we do produce one of these products. Touch a little bit about the types of ways we may test this, but just to give you more of the options that we may have when we look into testing these various stages of our products. So obviously the sequencing run is probably the end all, but then there's different ways that we could break up those fundamental components of the product and dive into how they may change with these various stress conditions. So one thing that could potentially happen is that you may have something that may appear on the surface once you have it exposed to high temperatures. Maybe something salts out, maybe there's particulates that start forming, who knows. So something like a microscope could be really useful in just checking the surface, doing that quick check, make sure that the surface looks like it should. Looks at least the way it started, or just minimal changes. The other potential option, fluorescence microscopy. So as we mentioned before, with sequencing, one of the big components of sequencing is that fluorescently tagged bases. And so if you're able to confirm that your fluorescence characterization on your surface remains the same before and after, then you're confirming that that starting point is going to be what you expect when you put it on the sequencer. So those are our fluorescence microscopes are really great ways to be able to do something that's similar to what we do on a sequencer, but be able to do it in a much faster manner. And then, of course, there are a long list of different analytical instruments and methods that we can use, like GCMS, HPLC, all of these various methods all provide us with other ways of really digging into the fundamental understanding of how maybe our surface changes, how a sequence or base pairs, DNA can change, all of these things that will give us better understanding before we want to get to the point of exiting development phase. So this gives you a general overview of how I can contribute into the Illumina process. Obviously, when you think about all of this is many weeks of group meetings and presentations, reviewing data sets, thinking about how to change different experiments and how we can move forward, and a lot of analysis at my desk. And you could think, me times hundreds of people that are all doing this, and then we're all compiling all of our data and bringing it together to create this package. And so we do this with every single product. And so I wanted to just kind of touch on how this was done a little bit with the NovaSeq X and some of the major areas that we were able to improve on in comparison to previous products that we've been able to introduce. When you look at the NovaSeq X, some of the main areas that we focused on in terms of what this product or what this product's line, how it improved from previous lines, was one, the cost. And so if you think back to that slide where I showed that graph of how we've been driving down the cost. So with the NovaSeq 600, we were right around $600 per genome. And now with the NovaSeq X, we've been able to decrease that to $200 per genome. And so when you think about how did you do that, like how are we still continually able to find ways to drive this down further? There's many components to it. One of the big things is the chemistry. this new chemistry that we introduced, it's more efficient, it's faster. And so it allows us to go through those sequencing cycles at a faster rate, at a higher quality. And so we're able to then provide this product at a cheaper cost. Additionally, there's been a lot of changes and updates to the flow cell, which has allowed more data, a tighter amount of data that has been able to be gathered and gathered on the surface of the flow cell. And then the instrument itself has many components, but from the optics, from the fluidics, and that have all helped to speed up those various sequencing cycles. And so these all contribute to being able to drive down this cost further. One of the other big factors has been the workflows as well. That has been a focus for the development of the NovaSeq X. So that included providing a more compact software package, something that's on board the instrument, something that you don't necessarily have to go and purchase separately. So that's been a huge plus of this new instrument. And that was its own effort of the software team finding ways that we can keep the footprint of the sequencer the same while also providing all of these new flexibility options to our customers. And then one of the most appreciated, I think, aspects of this new product has been sustainability. So as we all know, sustainability, caring about the environment, what we're putting into the environment, the type of waste we're producing has been a huge focus for our country and for the world. And so that's one thing that we've heard a lot from our customers of the amount of storage that's required, the packaging that's required when they purchase these large high throughput instruments, and how much that is a strain on their labs, honestly. And so we were able to condense the NovaSeq X packaging so that we're decreasing it by 90%. And so that allowed us to, as you can see from the picture, go from these many boxes to the single box. And that was mainly driven by the fact we were able to introduce ambient shipping, which was huge. So that basically, in comparison to previous products, we were able to confirm that we could ship our reagents, our cartridges, flow cells in a stable manner to our customers, so that when they receive it, no matter what conditions they go through, high, low fluctuations of temperature, long shipping times, that they're still receiving the same product that left our hands. And so that has been a huge plus. And we're hoping to just keep driving that further. I think the next area that I'm sure customers would love to investigate a little bit more is actual storage and long-term storage, which, that's on the marketing and customers' hands of when we're going to start trying to tackle that. But that gives you an idea of all of the areas that we focused on for developing and improving for the NovaSeq X. And when you think and compare it back to all of the other products that we've been working on, you can see how much this trend has gone in terms of data output and cost. And if you talk to anyone at Illumina, the goal is just to keep going. And so we'll see how that looks like in the next few years. But there's been a lot of just excitement about this new product and excitement about how we can continue to drive this innovation more and think about ways to ultimately get these types of products accessible to the larger community. Because as we continue to drive the cost down, that means that beforehand we may have had like really highly funded labs that had only had access to these tools, and so we would be really limited in terms of the number of experiments we could run, the number of data that we could collect. But now if we think about, okay, now we're talking about only $200 and this shorter amount of time that it takes to acquire all this data. So that just makes that window and that breadth of options much larger. That increases the number of countries we could potentially do a full human genome sequencing, the number of different populations we can gather data on, which then just provides us more information. Because the more information we have, the stronger our reference genome can become, the more access to a larger amount of variants becomes available, the better that we're able to create different treatments for cancer, for rare diseases, be able to provide information and guidance for precision medicine. Getting to the point of when you go into your doctor's office, you do a quick sequencing run, and then you're able to understand which medication you should walk out the door with. Like that's the general goal, and those are the things we're really working towards. And so hopefully in these next few years, we're able to get to that point where we're confident and able to provide these tools to the greater population. And so with that, I wanted to just give a quick shout out to some of the great engineers that I've worked with, engineers and scientists. And so this was the picture that was captured the day of the launch. And so they brought a NovaSeq X on site and had the photo ops that we could have had opportunities of. And this is just fun photography that one of my team members went and created. But obviously all of these products are not created by a single person. It takes many, many people to support the development of these projects. And I've been really fortunate to be able to work with some great people. I've learned a lot and have been challenged and have grown significantly over these years. And so it's been something that I've just been really excited to be a part of and be able to say that, hopefully down the road that this is a product that can help cure cancer, right? Like that's the ultimate goal. So thank you everyone for listening. Hopefully you learned something about Illumina and what I do. And I'm happy to answer any questions that you may have.
Awesome. Thank you so much, Obi. That was amazing. So we do have about 10 minutes, everyone, for some 10-15 minutes for some Q&A. And we do already have some questions in the chat. So if you do have questions, everyone, please post them in the chat and I'll just kind of scroll through as we see them in order here. But the first question that came up is, what is the most impactful application of this product in your opinion? I think that came up during the NovaSeq X slides.
I think the most impactful to me and what I really appreciate hearing about is at some of our company meetings is the applications has been those stories of where you see a child who has been going to the doctors for years trying to figure out what these symptoms are, what is it connected to, and it's getting to the point of you just don't know how much longer that life expectancy is, and then all of a sudden they have the option of here's sequencing, and they're able to understand what is different about that child's genome versus the reference genome, and then they're able to detect what they can do and understand what they can do. And I think those types of stories to me are just so impactful, because I think that just thinking about the amount of additional years that child may have, the limited amount of stress now that family may have, just the point of having an answer of what is wrong and what is ailing that person. I think those have always been the most impactful to me when I think about the various applications.
Awesome. Great. Yeah, this NovaSeq X is so exciting. This is going to be some big movements here. And I think in a lot of fields that just not only impact education, but like is going to change the way that we view the health industry and the way that doctors' appointments happen, treatments for diseases is so exciting, and how we can relate it to ourselves and our worlds. So the next question is, how have you been able to stay resilient and inspired in a role where you are constantly running up against challenges?
That is a very, very good question. I think that for myself, I think what keeps me inspired is being around people that are all passionate and excited about the technology. It always astounds me how much people will spend time, long days, just because they know that at the end of the day, if they can solve this problem, it'll make this much of an impact, make this much of a difference in the project. And so people are just driven by the need to learn and the need to, fix problems and find solutions and find new ways to get to those answers. And I think that honestly keeps me driven. The people around me, the people that these products could potentially help. I think all of that is what keeps me going every day. Because I know some days, you're kind of in the lab, you're in your little bubble, and it can be redundant in sometimes. But I think the end goal of what we're all pushing for is what keeps us going.
Yeah. So again, guys, we have probably 5-ish more minutes here for questions. So please throw some in. But I do have a question for you, Obi. What skills and or disciplines that you studied in your past do you feel provided you the most support, or where you gained the most knowledge to help you do the job that you're doing now? Was that a collection of multiple skills and fields? Is that, one particular subject, or internship experience, or, college class or something? What do you feel has helped you the most in the current position that you hold?
So I think for me, I will say that, all the areas and subjects I've studied, that those are great foundations to at least be able to understand that general knowledge of like science and math and physics and all of those things. But I think what has truly, truly helped me the most has been those experiences in which I've been in situations and groups that are very much like what I do day to day. There was the opportunities I had in college where I got to work in our biomedical classes, where we did designs and products together, did prototypes. We would meet, have our group meetings, talk about design, test it out, realize what was wrong and test it out again. And I think that those types of experiences have been really beneficial to me now, because that's honestly what we do all the time, is that we're just constantly trying to determine a design, determine all the ways in which it could fail, and improve on it further. And so those types of situations, being able to go through iteration cycles, being able to take feedback, really collaborate with people have been very beneficial to me to transition now into industry and take that into these various development products and projects that teams I've been a part of.
Awesome. Great. A couple more questions came into the chat here. So could you please elaborate on the output of this product? I believe the NovaSeq X. Is it limited to gene sequencing, or does it also provide analytics around protein activations, summary reports of potential diseases that an individual is prone to, things like that?
So this product is going to be more focused on high throughput gene sequencing, and so a lot specific to the whole genome sequencing reports and outputs. And so we're thinking of, humans, plants, animals. And so trying to understand, you can say the entire sequence and what that looks like. And so hopefully that goal will be to then use that data to direct it more towards like different diagnostic approaches, and as well as some medical treatments and things like that. Those will kind of be the main focus of this tool.
Awesome. Great. One kind of coming back to well, you kind of already answered that with your most exciting implications. Coming back to this other question, then we'll kind of shift back into this industry perspective. What skills do you see a need for in the industry right now? And maybe what do you see the needs being in the next 5 to 10 years in terms of maybe skill sets?
So I think a big emphasis in the last couple of years has been bioinformatics and software engineering. Those have been two areas that I foresee a huge need in the next decade, because as you think about all these ways that we're trying to increase accessibility, increase the amount of data that we're trying to gather, all of this, right, it's getting towards creating more data. And so when you have this immense increase in data, we need people to be able to analyze it, to be able to understand how we can continually have workflows set up in our instruments to handle all of this data. We have to think about storage. all of those things go into understanding how we're going to move forward once we have these terabytes worth of data in a single run. That is an immense amount of data that we're going to have to handle. But it gives us so much information, and so there is going to be a lot of focus and emphasis on really understanding it, handling it, and storing it.
Awesome. So yeah, the number of hard drives I have back in the day is external hard drives is crazy. But we'll end with this last question here. And then please everyone stay with us after as we'll talk about this room. But Obi, what advice would you give high school students who want to pursue a career in genomics? Oh, can you hear me okay?
Yes, I can hear you. Okay. So the advice I would give to high school students, I think it's a very good question. The advice I give to high school students is to start talking to people who are in genomics field and try and learn about all of the opportunities out there, because I think what can often happen is that when people think about genomics and research, they automatically think, okay, it's only scientists or only engineers that are there to support this research. But honestly, there's so many people that go into supporting this product. So when I mentioned that there's hundreds of people that are involved in a product, it's it goes everywhere from, yes, you have your scientists, you have your engineers, but you also have the supply chain who make sure that we are getting enough material in-house to make these products. You have the marketing team that is making sure they're talking to customers so they know that they'll be interested in this product. So you have the tech support who knows that they'll have an amazing background in science, but a lot of their strengths is like being able to troubleshoot with customers and communicate with them and then communicate back to us. And so I think that there's just so many different people that go into supporting this field. And I think that if this is something you're truly passionate about, find what works for you. You don't have to necessarily go to that science route, but if you do, awesome. We welcome it. We always need new minds. But if it's not specifically something you're interested in, but something you're super passionate about, find what does speak to you. Find what you do, where you want to see yourself moving forward. And I think that anywhere you kind of put yourself, as long as you're excited about it and happy to show up every day, that will be a great position to be in.
Awesome. Yeah, I think great advice, right? There's always going to be aspects of our jobs that, are hard to do, or it's like, oh, we don't want to do that today. But at the end goal of what you're working on brings you joy, and makes you excited. I think that was, yeah, absolutely. Thank you so much, Obi. This was wonderful. Before we have everybody, we conclude today's session. I did want to jump into one resource here. So if looking at, share my screen here. Can everybody see my screen? Looking at this DNA decoded page. So one thing I do want to share is that if you liked hearing more about what Obi did today, and learning more about her position, as well as want the opportunity to explore more careers in Illumina or in the field of genomics, and within this industry, I want to highlight this new virtual field trip. So here's what the webpage looks like. And if you scroll down, there's a multitude of videos here that are accompanied along with an educator guide, so interactive student activities that walk through and explore various careers within the industry and within Illumina specifically. And here's just a glance at all of the different wonderful staff at Illumina that explain what they do and why this work is so important. And especially as your students, for those of you in classrooms are, exploring careers. Another resource I do want to highlight here is also on the same page, but if we go to educator resources, here is a multitude of different lessons that touch on different applications of the field of genomics. This, as you heard here today, this is going to be a booming industry, field within this industry. And students are going to need skills if this is the aspect of the workforce that they wish to pursue. And so here's quite a variety of lessons here that range within 45 to 50 minutes to bring genomics curriculum into your classrooms. So I highly encourage that. And if you were, if aspects of today's presentation in regards to how the sequencer worked, and you want some more information about specifically bringing DNA sequencing into your classroom, we do have a really unique opportunity next week, a week from today, November 9th from 4:30 to 6:30. We are offering a professional development event for teachers on learning how to use a virtual simulation software that actually allows students to complete a DNA sequencing protocols and simulation lab. We will have resources available for this, curriculum aligned with this, as well as giving students the opportunity to perform research grade protocols and get some industry relevant skills and experience. So use the QR code right there to register. The registration link is also in the chat. Please join us next week, again, from 4:30 to 6:30. And with that, I will conclude today's session. I will be hanging around if anyone has any questions about some of the resources I just mentioned. And otherwise, thank you all for your time and spending the last hour with us. This was great. All right, and I'm going to stop recording here.